Hello, my name is Dr. Jessica Rutkowski. I'm here at the University of Illinois, and I'm going to be telling you today about my work putting predictive breeding into practice. I think as agricultural researchers, we've done an excellent job at improving production overall and yield, but we still have some significant challenges that we're faced with. And these are the profitability of farming and the environmental sustainability of farming. So how do we actually make growers actually earn more money from their hard work? And how do we make it possible to have high production and efficient agriculture while also mitigating and reducing harm on the environment? And oftentimes we think of these things as having to make a choice, between profitability or sustainability, but how do we actually achieve both? And here in the state of Illinois, this is a significant problem for us because we're one of the states that contributes most heavily to the nutrient runoff that goes right down into the Gulf of Mexico, and we're one of the worst contributors to the dead zone. And we really need to come up with ideas on how do we reduce our impact in this way while also maintaining the profitability of farming. And I think small grains, especially winter wheat, is a good part of the solution because not only does wheat contribute to profitability in the cropping system, but it also can help it reduce the impact on the environment by covering the ground over the winter and preventing soil erosion and nutrient loss. Now, normally you don't think of Illinois as being a major wheat producing state, but in the eastern US, we're actually one of the most significant wheat producing states compared to all the surrounding states in our region. And we actually have currently 940,000 acres of winter wheat planted in the state. Most of those acres are concentrated in the southern part, where we can be grown in a double crop system very, very effectively. And with that system, how it works is you plant the wheat in the fall, and then in the summer, immediately after the wheat is harvested, the soybean is planted. And this has really enabled the state to be a significant wheat producer in the eastern soft red wheat growing region. I find it very encouraging that this system is profitable, and it tends to be profitable across years, and especially when we talk about Southern Illinois. Here's just a graph of gross profitability of the wheat soybean double cropping in Southern Illinois from the years 2016 to 2021. In the blue dotted lines is the wheat double crop soybean, the combined gross profitability of those two crops. And the y-axis here is dollars per acre. The green is just soybean on its own, and the red is corn. And gross profit here is just the total profit, total revenue from the yield times the price of the crop minus the cost of production. And so for me, what I am focusing on is how do I improve this further? How do I make this wheat soybean double cropping in Illinois and the surrounding states even more profitable and even more effective so that more people can benefit? And then potentially we can even expand this into other areas, such as in Northern Illinois. And I'm doing this as part of my group, my research group of small grains improvement at Illinois. And we are a combination of two research labs working on small grains. I am leading the winter wheat breeding and research, and Dr. Juan Arbalez leads spring oats. And so we we all work together as a team. We have a lot of graduate students and undergraduate workers and a senior research specialist. And together we work together on how do we improve small grains for our region so that we can help contribute to sustaining people and the environment. Now, I'm relatively new here at the University of Illinois. And when I came, I was very fortunate to have inherited a strong breeding program with really consistent genetic progress. And based on 20 years of data that we analyzed, this work was actually led by one of my graduate students, Lucas Winaro. He analyzed the data across 20 years from 2000 to 2020 and looked at gain due to genetics in different traits such as yield, test weight, height, days to heading. And we found really favorable results in all the traits of interest. Particularly, I want to highlight the trends we're seeing in grain yield. And we found 0.5 bushels per acre per year. So half a bushel per acre per year is what we've been able to achieve. On the map here is just showing you where the testing sites were in this range of years. And so you can see we're sampling the region where mostly wheat is grown. In the south, those orange dots correspond to the acreage of wheat. And so on this plot here, you can see this trend going up, and that's the slope of this line is 0.5 bushels per acre. So on the y-axis here, we have bushels per acre. On the right, kilograms per hectare. On the left, the x-axis is the year. And this is encouraging because it tells us the program is worth investing further in. We don't have major issues with the operation of the program itself, or some really problems with the germplasm, or it's not that we don't have variation. We have a good solid foundation to build on, and I'm really grateful for my predecessor for leaving that with me. But in spite of that success, I came to Illinois wanting to take all the things that I had learned at the various places that I have worked, and I wanted to actually be able to make changes that I had never been able to make before. I had never been in the position to actually make change in a breeding program, and that's something I really wanted to do. So I came to Illinois wanting to make changes and wanting to make improvements, and that was something I just was so eager to do. And so I took the conventional breeding strategy, and I basically started from a blank sheet of paper and wrote down what I think the strategy would look like. And I'm still actually achieving that, but I've come pretty far in achieving that ideal strategy. And this is what I have settled on currently. This is our new breeding strategy, and really it has three main activities in parallel, which is overall it's pretty standard. Those three main activities are quite core essential activities to any breeding program. And so that's recombination and line development, phenotypic evaluation, and pure seed production. But the exact details are probably different, and definitely different from what they were. So I'm just gonna focus mostly on the recombination and line development and on the phenotypic evaluation part in this presentation. And I'm going to start with that recombination and line development and walk you through a little bit in more detail what those steps are. Before I get into all those nuts and bolts, first, I want to talk about the objective and how I frame the objective in the breeding program. What I had always wanted to do is select based on total net merit. So that's what I decided to do. Total net merit is actually profitability itself. And that's all, since that's ultimately what I want to maximize, I want to select directly on that. So what I do is I take all the traits that are economically important: yield, early maturity, deoxynivalenol test weight, and I use that to compute the potential profit of each and every line if that line were to be grown as a variety in a farmer's field. And so I have a function that I can plug these values in and generate a value for net merit. And then I use that when I'm making selection decisions, especially for parent selection. And just to explain a little bit further, what does this function actually look like? So we have yield in bushels per acre times the price per bushel. So that would be, the total, gross revenue. But then we have to factor in some other things. So first, the price will depend on the quality of the crop. So how much dawn is in the grain? What's the test weight? So those traits affect the actual price. And then we have to factor in what's the soybean profit that we would gain due to the wheat being earlier maturing. So we factor that in because we know that for every day that we can plant soybean earlier, we can improve its yield by a half or even a full bushel. So that has a significant impact on the profit. So as I said, I use that a lot, especially for selecting parents. But then after selecting parents, I have to actually decide what crosses to make. So I do that by taking every possible cross combination and making a ranking based on its net merit. It's like I predict what the average progeny will be, and then estimate the net merit of each progeny. And then I came up with a simple, shiny app that runs on an algorithm in the background that allows me to rank those on the fly, so that when we go into the greenhouse, I can pull this up on the tablet and I can enter in everything that is ready to be crossed that day, everything that is could be emasculated or used as a pollen parent. And then I type, I can actually go in and type those in, and it'll tell me what's the best list of combinations I can make that day. So it's really nice to make things more efficient and be really efficient with time and get the best possible crosses. So that's just a kind of a fun thing that we've been able to do and streamline our crossing. Once we do make the crosses, now comes the actual line development. So that's where we take those seed from the crosses, we plant F1 plants. Seed from those F1 plants are F2 seeds that goes into F2 bulks. And we're advancing them in a kind of speed breeding protocol that was developed by MSU, and we've taken it and adapted it to our conditions. And so what we do is we vernalize these into these plastic containers. Actually use a long vernalization time for this because we found that there can be a lot of competition within those bulks if some of those plants don't vernalize well. And our germplasm tends to take a long time to vernalize. So then we take each of those, after 10 weeks, we take each of those bulks and put them into a pot in the 22-hour light greenhouse. And within less than 2 months, we have our bulks reaching maturity. So we take those F2 bulks, we harvest the F3 seeds. We get those F3 seeds in November. And now the question is, what do we do with that in November? It's too late to plant it in the field. So we have some choices to make. We could advance it another generation and then plant it F4s into the field perhaps, or we could try a couple other things to try to get to a fixed line even faster. So these are the ideas we're currently now testing. So we've sent our F3 bulks to a winter nursery in Yuma, Arizona, wherein we're going to select single spikes out of there, bring them back to Illinois, put them in hedgerows, then take those hedgerows and put the selected hedgerows into small increase plots. That helps us, that whole system with the speed breeding and the winter nursery helps us save a year. But then we're also testing a different approach, which is to take that F3 seed that we get from our bulks and start vernalizing them in our cold room in a tray, and then take that and transplant it into the field in the spring. And so that is nice because that allows us we don't waste any time. And also we have the ability to space plant these plants in the field so that we can get as much seed as possible. In the conventional system, we're taking single spikes, so then you're not getting that much seed, but you can get a lot more seed from an entire plant. That lets us increase the seed 1 year earlier, which is great for getting it into testing sooner and just speeding up the whole pipeline. It's a little bit trickier pulling this off, but the time savings is worth it if we can make it work. This is work that my graduate student, Jeremy Lograno, is working on and taking the lead on. So I wanted to share you some pictures, how it's looked like. We tried this for the first time last season, learned a lot, and now we're applying the things that we learned and doing it again this year. So here's the system we're using for the transplanting. If you've been wondering that, we put them in a paper pot transplanter that was actually designed for vegetables, and then we vernalize them in these trays in our vernalization cold room. We have multiple families here planted in a tray, and then we use this transplanting device to put them in the field in the spring. And here the picture on the right is showing you the picture of our first single plant nursery, and we have, as you can see, irrigation there to make sure that the plants are fine after they're transplanted. So next, I want to focus in on the evaluation system and some of the things that we've done to try to improve in that area as well. For our yield evaluation, we use 5 major locations within Illinois where we have a large number of our breeding lines being tested. And then in addition to that, we have acquired 8 out-of-state locations through different collaborative agreements with other public breeders. And I have to say that the wheat breeding community is extremely collaborative and open, and that's been a really, really nice experience for me. It's been really nice because I can actually make suggestions and come up with ideas and propose things to different people, and most of the time everybody's on board, and it's like such a nice way to work. So I think what helps a lot, I'm getting a little sidetracked here, but I think it's important. What helps a lot is we trust each other, so we know and we reciprocate. So if somebody does something for someone, then we that favor is going to come around in return back again. So we're very collaborative and open, and I think that really relies on that reciprocating and just being a good team player. So that has really helped us expand our testing. We also, I want to mention too, we do a lot of testing on farmers' fields, so we really only have in our testing here in Illinois, only 2 of these are on research stations, and a lot of that has been out of necessity because we don't have a ton of research stations here in the South that we can use. But we also tend to find that the data we get from farmers' fields is just as good, even a lot of the time better. than the data we get on our research stations. So that's been also a really nice value that we've gotten here in our yield evaluation. Here's an overview of the testing design that this is what has been planted in the fall. So this is very up to date. Here on the along the from top to bottom here is the names of our different testing locations. And across the bottom are the lines that we're testing. So, and then the color coded is corresponding to replication. So lines that are not replicated in a particular location are coded in orange, and the lines that are replicated in a particular location are color coded blue. So we have a little bit of a sparse design, although not completely following a sparse testing strategy as of yet, but we're getting there. So we have stage 1. We have our lines being tested in one location, either Urbana or Neoga, just about an hour south driving from Urbana. So similar environments, but a little bit different. And so that is an unreplicated single location. And then our next stage of testing, so as we advance, we select lines from stage 1 that move to stage 2. Now we have these at 4 locations, and there's still very much partially replicated. So we have lines, only a proportion of lines being replicated in a given environment. And then we try to make sure that overall, the same number of plots is allocated to each line. Then, as we move from stage 2 to 3, we're adding another 3 locations. So 1 additional location in Illinois, and then 2 locations in Virginia. And same principles with the replication. It's partially replicated. And then from there, we select some lines that move to stage 4. That's our kind of final stage of testing, where most of the time, in most of the environments, it's replicated twice, with some exceptions here with our out-of-state locations, where we have a limitation in the number of replicated entries we're allowed to send according to that agreement that we've made. So a basic idea here is, as we move across from stage 1 to 4, we are applying partial replication and then only really replicating fully in each location at the very latter stage testing. What I like about this is we have a very good connectivity across all the locations. So the stage 4 has been tested in the past in all these locations, and that's going to connect across everything. But those stage 4 lines have also been tested in previous years, which helps connect across the years. So that is really a good feature of the design. And in addition to having that connectivity through those common lines through the environments, we also, because we're using genomic selection now and genotyping all the lines, we're using that when we analyze the data, so that now our connectivity is not solely based on just lines overlapping, but in addition to that, having marker data and relationships between lines that overlap across environments. Now, we don't want to miss out, of course, on everything that's high throughput phenotyping can potentially give us. So we're investigating and exploring the idea, the potential to use aerial high throughput phenotyping using drones to help us improve our genomic selection models. So we've come up with this idea that we're testing. It's called phenomic assisted genomic selection. And the concept here is where we would actually plant twice as much as we would normally be able to harvest. And with the intention that we're not going to harvest all of it, of course, because we do have limited labor time and equipment at harvest. And so that we would take our HTP data to fly the drone throughout the season at those locations, and then harvest a subset. So we'd harvest what we call the core training set, which would be our typical lines that we would normally test. And then, but remember, the HTP data is on everything, which is our core plus what I'm calling an accessory training set, which will not have real yield data. Then with those two pieces of information, I will use that to impute grain yield on the plots that we did not harvest, and then use all of that data in a genomic selection model. So in a sense, we're using kind of predictive yields to help train a model to predict yield. Sounds a little bit crazy, but this is something we're currently working on. I think within the next year or two, we'll have some interesting results to share on that. And this work is being led by one of my PhD students, Heisa Bartowski, who's in the picture here. In addition to yield, we've done some work on improving our scab resistance evaluation. So as I was mentioning earlier, deoxynivalenol is an important trait for us because it can affect the final price of the grain, which affects profitability. Deoxynivalenol is caused by the pathogen of Fusarium graminearum, which causes a disease that we call scab. So essentially, we want to improve scab resistance to reduce DON and reduce the impact that DON has on profit. So we have been doing this in the program since before I got there. So the program has a very good understanding and protocols and has been very successful in improving scab resistance, but it has been a huge investment, very labor intensive, and a lot of resources go to this. So I wanted to come up with a way to make this more efficient so that we can invest resources in other things that really needed more attention. Now, the challenge with scab resistance is it's a quantitative trait with low to moderate heritability. And so that really makes things difficult for us. It's not a simple disease that we can just improve and move on. It's something that takes a lot of time incrementally and a lot of resources. The conventional phenotyping approach for scab resistance has been, to use an inoculated and misted nursery, typical of a lot of other diseases where you would need to do that. But we've also had to evaluate a lot of different traits and so go in and rate these symptoms throughout the season. So for example, incidence, we have to go in and rate each plot for the proportion of, percentage of spikes within a plot that have any disease. That's incidence. We have to look at severity, which is the spread, the percentage of the spike that has symptoms. And then the worst of it all is we have to actually harvest each and every of these rows and thresh out the grain, look at the kernel damage, and estimate the percent kernel damage. And then even worse, we have to grind up these samples and send them off to a lab and get DON data. So you can imagine all the steps and all the people and all the labor that's involved. And because of that, because of the grain phenotyping is so onerous, oftentimes breeders rely on just indirect selection using these visual symptoms in the field, which is, we know indirect selection is okay, but it's not as good as direct selection. I should also just say that the program, because of the low heritability, the program had been doing 3 replicates of each line in the nursery, and in total scoring a total of 5 traits. So it's a lot. It's a lot of data. So I wanted to try to find a way to, make this more effective. Can we use multi-trait genomic selection to our advantage to make this breeding for this trait a little bit more efficient. So now that we're genotyping everything in the program and using GEBVs for selection routinely, this opens the door to actually use GS for scab resistance without even adding an additional cost, because we know we want to use it for traits like yield. So what we did to investigate how to use multi-trait genomic selection to our advantage, we looked at testing some different models. So we wanted to ultimately find out what traits, what's the minimum number of traits we need to phenotype, and do we really need to phenotype all of the 5 traits? Can we get away with doing less? So we looked at using this approach where we have a training set, and we have the training set has marker data. It has all the phenotypes for traits other than our primary trait, which is DON, and then it has our DON data, our phenotypic data, and that's something that we have to have in the training set to be able to train the model to predict DON, which is ultimately what we want to predict. And then with our new breeding candidates, we have their marker data, and then we have phenotypes for traits other than DON, and that's going to help us predict their DON data, DON phenotypes, or breeding values, I should say, so that we can actually predict before we have data on DON. Right, and so this is some of the results that we looked at to try to determine what should we do to improve our efficiency of scab evaluation. This was work that was done by a postdoc that was in my lab, who has since moved on to Bayer. So he looked at this, and he actually looked in 2 different data sets, one from the University of Illinois and one from Purdue, where he got his PhD. So that was nice that he could, and because we being a collaborative community, we could actually join forces and look at across 2 different data sets. So here on the y-axis, we have predictive ability of the different models or prediction approaches. On the x-axis, we have all the different ways we tried to predict DON. So all the way to the left, we have different phenotypes that we use to predict DON. The far left is ISK, which is an index of incidence, severity, and kernel quality index, which is same as FDK. Then the one to the right, that's fusarium damaged kernels, the percentage of the kernels that have fusarium damage in a grain sample. And how did that predict DON? And then the third bar here is a single trait genomic selection model for DON, so like just a typical univariate model for to predict DON. And then we have all the other bars here in our boxes here in the box plot are all these different multi-trait genomic selection models with different combinations of traits. And what we're trying to pick out is which one of these is where you get the most bang for your buck, or like the best accuracy with sort of the least amount of traits. And what we kind of honed in on here is that it looks like fusarium damaged kernels and DON together in the model are really sufficient to train an accurate model to predict DON. And of course, it's better than just univariate model with just DON alone. And you can kind of easily see if you look across, like see, I pointed out with the yellow arrow, that particular model. And if you look across, you don't really find any other multivariate models that are much better than that. And so that's really the sweet spot where we can get the most bang for our buck. Of course, another interesting one would be to just look at, what would those visual symptoms, if we just did visual symptoms together with DON, what would that look like? And you can see it's it's kind of lower. It's unfortunately not um, extremely high. So, it seems like if we could just get a good estimate of damaged kernels and done, we can train a model and accurately. And one thing I also want to point out as a comparison, if you look at this model accuracy or predictive ability compared to the phenotypes alone, you can see it does improve upon if you just have phenotypes alone, which is great because it's like we are adding to our accuracy by just increasing our data, like using our previous year's data on Don, using our marker data that we already have, and really just increasing the accuracy compared to that. That's in the Illinois data set. Now, if you look at the Purdue data set, the story is a little bit different. This model that has FDK and Don and marker data to predict Don in our candidates is actually having less predictive ability than the model, than just the phenotype alone, which is definitely not expected. And we determined that in this Purdue data set, the design that was used in the field evaluation was not really amenable to being able to fit this kind of model. So the problem here is we're not getting a good estimate of our variance components. It was a kind of an augmented design with very few checks. And so that might be great for a conventional selection approach. But if you want to try to fit a model like this, the design has to be different. So that's a good lesson to learn, is that if you're going to use these kind of approaches where you're fitting genomic prediction models, especially multi-trait models, the design is going to have an impact, and you have to take that into account, or you have to make sure you're making those appropriate changes to your testing designs so that you can actually take advantage of these methods. So I've kind of taken those things that I've learned through the work that we've done and applied it in the program to improve our strategy. So now when I'm evaluating scab resistance in our Illinois breeding material, I'm only looking at kernel damage and Don. So I'm really, unfortunately, I cannot avoid, harvesting the grain sample and cleaning it and grinding it, sending it for Don testing, because I do need to get good data on that to train the model, of course. And I do actually use data. I use in our early stages of testing, I do not have Don data on those lines, and I can just use kernel damage and my prediction model to predict Don. So that's been very helpful at getting a good accurate selection early on. And I want to mention too that I've also looked and found that I can reduce replication within the nursery and still get very accurate selections. So here on this box plot to the right, I just want to show you my selection accuracy. And I've looked at this selection accuracy for Don before I get the Don data. So that's critical for me because I don't get that data before planting. And with the accuracy, when I have 3 reps, is here shown on the very far left of the chart. And then when I have 2 reps, but I'm using genomic BLUP to make the selection accuracy is actually on average a little bit higher. Of course, the best accuracy is when I have 3 reps and I use a genomic prediction model. But the advantage of that is pretty small, and I think not worth the additional resources that would be needed for that additional rep. So that's kind of the sweet spot I found on replication. And so that has now framed my strategy where I have this reduced replication, evaluating only the kernel traits like kernel damage and Dawn, and then using, of course, multi-trait genomic selection every time I do selection. Now, to take that even further, to try to make this even more efficient and easier, perhaps more accurate, we've looked at, can we use actually artificial intelligence to evaluate kernel damage? So instead of looking at a petri dish of grains like we see on this slide here and just estimating visually what's the percentage of the kernel damage, so that would be the conventional way of doing it, can we actually use just take a picture of the grain sample with a handheld, like a cell phone that we use to collect data, and then use that for phenotyping? And can we use, technology, the same technology that Facebook uses to tag people in photos and identify objects? Can we use that, but for the purposes of phenotyping? So this is something that we've been working on, and actually the papers recently got out on this, and we've had some pretty encouraging results. But first, I want to walk you through a little bit the phenotyping approach, because it's a little bit unique. So what we do is we take these petri dishes of grains. That's normally what we do when we're visually phenotyping. We pour that in a petri dish and rate it. But this time we take a picture of it with our cell phone camera. And we have these raw images. You can see it towards the very left. What we have to do then is label each and every kernel. And that means tracing around the boundary. So we have to train the artificial intelligence what a kernel is. It doesn't know what a kernel is. And then we have to train it what's a diseased kernel and what's a healthy kernel. So those are what we call our true labels, that tracing around each of these kernels. That's true label. Using true labels, we can calculate FDKL, fusarium damaged kernels, from labels. That's what that stands for. Using the true labels, we also train our artificial intelligence model, which is called mask RCNN. And then we deploy that to a new set of images. So here is, again, a raw image that we take with our cell phone. And then that mask RCNN is applied to that, and it predicts labels onto those kernels using the training that we provided it. Using the predicted labels, we then calculate FDKL hat, which is our fusarium damaged kernels based on predicted labels. So this is something that we've done and evaluated now. And for our very first attempt at doing this, we have used 659 labeled images, which is actually very small in the artificial intelligence world. And so, it's a pretty difficult task, actually, to be able to train a model like this with a small number of labeled images. We went ahead and we did it, and we looked at how effective it is for selection and see, are we on the right track? Does this make any sense at all? And what we found was that our genetic correlations with Dawn, so taking those kernel damage values, and getting a genetic correlation with our Dawn, our primary trait, with our predicted label. So what mask RCNN told us FDK was, it's the correlation with Dawn is 0.58. With our fusarium damaged kernels, our FDK, from visual estimates of people looking at the kernels, the genetic correlations with Dawn are higher, 0.84. So it looks like humans are kind of winning here, which is not shocking considering this is our first time with a very small sample. And, just to confirm, if we feed this into a genomic selection model, what's the accuracy for Dawn? So just by comparison, in this data set, if we just use a univariate genomic selection model for Dawn, accuracy was 0.26. If we use the FDKL from our artificial intelligence, it's 0.61. And if we use our visual ratings, our FDK visual, FDKV, that's 0.97. So you can see that basically having that FDK from the mask RCNN, what was predicted from our artificial intelligence, it's better than having nothing, but it's not better than a human being. So we still have a ways to go, but we're getting there. And so what we're currently now in progress, in the process of doing, is adding more data. We have more than twice as many labeled images ready to go that need to just be need to be fed into the model. And we have a lot of other unlabeled images that need to be fed into the model. So we have a lot of potential to improve. And ultimately what we want to do is develop a simple tool that reading programs can use. So some kind of a web portal where you can upload your Scab images, kernel images, and get some values out that will completely take away the drudgery of having to evaluate this trait and the time sink that it takes at such a critical stage when we're trying to make selections between harvest and planting. It's such a short time frame, very stressful time. So this would really help in making that a lot quicker and a lot easier and even standardized and less subjective. So I just want to leave you with some final thoughts, kind of what I've learned through my experience working with trying to implement predictive breeding methods in a breeding program. I think what I've learned is just to take the plunge and just do it, because doing it is how you actually learn how to do it better. And you have to actually try and learn and work through the issues that you find as you go. And so sometimes you don't know all the things that could possibly happen or go wrong, but you have to just do it anyways. And I think sometimes we test things a little bit too much before implementing them. And so we need to move. Sometimes I think we just need to stop testing things and just do them. And that's kind of my message. And something that I've learned, or my, kind of my point of view, I want to leave with all of you today. With that, I want to thank everybody that contributed to this work. For the Fusarium work, I want to especially thank Rupesh Gayer, who is the postdoc who started the work, and then Girish Chowdhury and Junzi Wu, who did a lot of the heavy lifting and all the work on the artificial intelligence. I want to thank my graduate students, Lucas Minaro, Jeremy Lobrano, Heisa Hebertowski. They've really made it possible for me to make all these changes. And I didn't even get to tell you all the other changes that we made that they helped me do. So that has been really exciting to see that. And it's been a lot of fun. And then I want to thank especially Tadele Kumsa, who helps keep the program running from day to day, and Darren Joes, who has been helping us a lot in our how we carry out trials, and in helping us improve in that dimension, and a lot of helping us a lot with field work too. Also, AJ Ackerman, who took the Scab phenotyping work and finished it up and got it so that the paper could get out, and added some additional analysis to help evaluate how the models were performing. I want to also acknowledge Milka Kagoni and Anoop Jakal, who are graduate students working with Dr. Juan Arbalais. And with those three, they contribute a lot to helping the program be successful, and providing some useful comments and helpful hints. And then I also want to acknowledge all the wheat breeders in my region, over here on the right, who've helped me a lot with, by being good collaborators and participating in the multi-state testing design that we put together. And in addition to that, of course, all the sources of funding that are listed here, I want to make sure to thank all these that have contributed, especially the Wheat and Barley Scab Initiative has been one that has been very consistent throughout the years, and definitely a huge contributor, as well as all the others here on the slide. Thank you to them. So that's it. Thanks everybody for listening. And please, if you have any questions for me, you can always reach me by my email, jrut@illinois.edu. All right, take care, everyone. Goodbye.